This quarter, we learned about the renal system in humans. The renal system consists of the kidneys and is used primarily to regulate blood volume and pressure, eliminate waste, and control electrolyte and metabolite levels in the blood. It does this through a complex system of blood filtration and solute reabsorption. Throughout each unit of the kidney, known as nephrons, and into the collecting duct at the end of each nephron, the kidney uses a compound known as urea to set up osmotic gradients that favor the reabsorption of water and nutrients into the body from the fluid in the kidneys. As we discussed this in class, I found myself wondering where in the body urea comes from. We had discussed where the various hormones involved in the kidneys come from, and we discussed the origin of the molecules being filtered, but we never discussed the origin of urea. As I began to look into this, I found out that urea contains nitrogen, which is primarily produced by the body from the catalysis of proteins. However, when proteins are broken down, the byproduct is ammonia, and not urea. As I looked further into ammonia production, I found out that ammonia is very harmful to the body. As ammonia builds up in the blood, it can begin to damage various tissues, starting in the liver and kidneys, and eventually the brain. Prolonged high levels of ammonia can even lead to encephalopathy, which almost always leads to death if untreated. Treatments include taking medications to remove ammonia from the blood, limiting protein and nitrogen consumption, and some forms of dialysis. In extreme cases, a liver transplant may be the only way to save a patient's life. In order to prevent the damage caused by excess ammonia, the body converts ammonia into a less harmful form right after its creation and excretes this new form through urine. This conversion, known as the urea cycle, takes place in the liver. For every 2 molecules of ammonia, the liver uses 1 molecule of carbon dioxide, 1 water molecule, and 3 ATPs to produce 1 molecule of urea. Compared to some of the other metabolic cycles covered in class, this may seem like a lot of energy simply to produce 1 molecule, but we must remember that the body is trying to quickly process ammonia before it can cause any harm. Before we go into the details of the cycle, here's a quick overview. The production of urea from ammonia is a 5-step process using a variety of enzymes. The first 2 steps take place within the mitochondria of liver cells, while the final 3 steps take place in the cytosol of the same cells. The first step of the urea cycle consists of the enzyme carbamoyl phosphate synthetase using 2 ATP to combine the ammonia and carbon dioxide into a single molecule, carbamoyl phosphate. From here, the enzyme ornithine transcarbamylase combines the amino acid ornithine with the carbamoyl phosphate molecule to produce the alpha amino acid citrulline, which passes out of the mitochondria into the cytosol of the cell. Citrulline is then bonded to another alpha amino acid, aspartate, by argininosuccinate synthetase using 1 ATP. This produces the argininosuccinate molecule. This molecule is broken into arginine and fumarate by argininosuccinate lyase. The final step of the cycle consists of the hydrolysis of arginine by arginase 1, producing 1 molecule of urea and 1 molecule of ornithine, which re-enters the mitochondria to be reused. From here, the urea is transported to the kidneys, where, as discussed before, it is used to set up osmotic gradients and eventually excreted as nitrogenous waste in urine. As I was researching the urea cycle, I learned that it was actually the very first human metabolic pathway discovered. It was discovered in 1932 by Hans Krebs and Kurt Hinseleit, 5 years before Krebs discovered what is now called the Krebs cycle, also known as the citric acid cycle, which is the phase of cellular respiration that produces the majority of the ATP from glucose. Krebs' work on the urea cycle was based on the discovery in 1904 that urea could be produced by the hydrolysis of arginine. Krebs realized that a slice of liver, treated with the appropriate reactants, could be used to synthesize urea from ammonia, and used his analysis of this to explain the urea cycle. Krebs' work on metabolic pathways, using live tissue samples, set the groundwork for many future discoveries of metabolic pathways, including, but not limited to, the citric acid cycle, glycolysis, amino acid synthesis, and fatty acid metabolism. In 1953, Krebs was awarded the Nobel Prize in Medicine for his contributions.